Okay. Now I am going to present the workflow and the details for the analysis of microarray data. And this microarray data could be for gene expression, SNP profiling, or any kind of high throughput data analysis. So here I will explain everything step by step for microarray data analysis. And the steps includes here are sample preparation, hybridization, scanning, data normalization, statistical analysis, biological interpretation. So sample preparation will include collection of and preparation of biological samples, labeling and binding of samples to the microarray, detecting hybridization signals, adjusting for technical variations, then to identify significant genes in terms of expression, mutation, or epigenetic modifications. Finally, understanding and interpretation of the data in terms of biological interpretations. In general, in sample preparation, it includes extraction of RNA. For example, this is for gene expression purpose. Then go for preparation and labeling of samples, hybridization. Labeling of samples could be with the dyes like Psi3, Psi5, then go for hybridization, washing, and finally image acquisitions. So in hybridization, here you could see targets are isolated and labeled. Then go for labeled targets which are combined with array. Then array is washed after hybridization, and finally go for hybridized array scanning. So in the next part, scanning. Scanning includes like image processing, spot identification, hybridization, intensity measurements, background subtraction, or denoisation. So then go for next step, signal intensity values for each gene or target. And finally, go for significance of expression values, where you could measure, for example, fold changes, p-values, and then both the things are considered for considering the genes as differentially expressed up or down regulated genes. Finally, go for biological function analysis or gene set enrichment analysis, exploration of data, and result validation by using further experimental approaches. Here, data normalization. This is just a kind of workflow which could be seen here clearly. Go for image processing. Well, this looks like in this way, but when you are using some software packages, for example, in MATLAB, then you need to download the raw data files. It could be in different formats, for example, .cell format, then download the library files from the main source website or also some other software pipelines and extract the probe and probe intensities and then go for normalization purpose where you could use GCRMA, RMA, or EB approaches. So this is what you could understand. And then finally, after you normalize the data, go for denoisation, everything, then you generate the proper expression values for each probe. Then each probe needs to be annotated in terms of gene. So all the probes IDs could be mapped for respective genes, and then each gene will have expression values. And finally, once you have the expression values in different experiments, then you go for the comparative analysis in order to compare whether the genes are upregulated or downregulated. And after you calculate the comparative analysis between two or more than two conditions, for example, the healthy human samples with respect to cancer samples. Then you calculate the fold changes. After you calculate the fold change, then you go for p-value calculation, or you can first calculate the p-values for each gene, then go for fold changes calculation. After you calculate all these two values, p-values and fold changes, then you assign a threshold level of for fold change as well as p-values, and p-values must be less than equal to 0.05 in order to consider it as significant. Fold change could depend, but in general, the people use plus minus 2. That means when the fold change is equal to or more than 2.0, that means the gene is upregulated. If the fold change is less than equal to minus 2.0, then the gene is down regulated or down expressed. So here you will have two values for each differentially expressed genes, but fold changes are not kind of fixed things. It will depends. For example, in human cancer, the fold changes are mostly quite high. Then if you want a strict parameter in order to consider only highly significant values, then you could also increase the fold change length, like threshold, like plus minus 3.0 or plus minus 4.0 or plus minus 5.0. It depends on how big ranges of fold changes are present. Now, here you could go for heat map plotting of all the significant genes or the genes of your interest. And also you could go for clustering for all the samples based on expression values or based on fold changes. And finally, here it starts to go for functional annotation, for example, gene ontology, KIT pathway analysis, or many more things. So there are some of the typical analysis, for example, heat map plot with clustering or group classification or class discovery. Then it is like the intensity versus array, how much variations are present between different kinds or conditions of data sets. And then, for example, here, D is representing volcano plot, where you plot the genes with fold changes and p-values in one plot. And next one is p-value distribution, which you could see here. And if you want to see the details, then you could also see the referred paper, that is Here cited Nature Reviews Genetics. Nature Reviews in Genetics, and was published in 2006. So the right side dis- displays the classification of the samples or the genes. For example, in class discovery, and the classification could be supervised, unsupervised. In supervised, you could also go for a statistical interference or inference supervised learning. Class discovery, in case of unsupervised learning, here you could identify molecular phenotypes such as oncogene specific classification, discovery of cell of origin in cancer profiles, finding human like mouse models of cancer, then go for a statistical inference in supervised learning, identification of differential expressed genes, go for tumor versus normal tissues, changes in gene expression levels, and then gene expression changes during cancer progression or different phases of cancer. Then go for class prediction in supervised learning, go for cross species prediction of tumor phenotype subtypes, prediction of the responses to therapeutics, go for prediction of a functional gene sets. So these are some of the mostly used targets for such microarray data sets. And the same thing could be done for epigenetics analysis also, or mutational profiling. Now it comes to biological interpretation. As I mentioned, it could be gene ontology or gate pathway or any kind of biological function, relevance or inference of for each gene. So here you could also go for more analysis like co-expressions of genes or some kind of correlation analysis. And these are some of the examples how you could go for different kinds of biological interpretations. And then this is overall thing, but you need to remember a few things. There are some of the pipelines, inbuilt pipelines, which are freely available. For example, in R packages also, MATLAB also, Python also, where you can just go for uploading the samples and most of the analysis it could be done. But there are some, like if you are downloading the publicly available data sets, then for almost all the microarray data analysis, there is inbuilt software which automatically analyzes for different groups, and the groups you can assign, and they will generate the differentially expressed genes with fold changes, p-values. And next, a part of analysis, for example, biological function analysis, you could do it by using other software, for example, David or EnrichNet or Panther databases. So this is overall, in summary, what you could do for microarray analysis. But before you go for analysis of any microarray data, please read the relevant research and review papers, which will be more helpful for data interpretation and analysis. Thank you very much.